Hi, my name is João Rosa, and I'm the head of sales of scientific instruments at Insplorion. Today, we're going to have a look at Insplorion, where the company comes from, its technology, and the applications that can be useful in these post-COVID times. So, who are we? Insplorion is based in Gothenburg and was founded in 2010 as a spin-off of Chalmers University. Our core is on the nanoplasmonic sensing technology, which we call NPS, which is a technology that is verified, patented, small and robust, but also sensitive and scalable. We've been working with our scientific instruments since 2010, and they've been commercial since 2011. And right now we have three main products, X1, the X Nano, and the Acculite. We will be focusing our presentation later on these instruments. However, I think it's good to mention also that there are other platforms that we've been able to develop based on the same technology. One of them is the gas sensors, which has been active since 2016, which is a common platform that is gas specific for sensor chips. And right now we're working on nitrogen dioxide and also on hydrogen. The other platform is a battery sensor, also active since 2016. That is a fiber optic probe inside of a battery cell that will allow us to also monitor the chemistry and the temperature within the batteries. Our team is composed currently of 12 members that were well spread between sales and marketing and product and technology. We have a team of extremely dedicated employees that are a great combination of entrepreneurship and technical competences with experience also in sales and marketing. On the technical side, Dr. Elin Langhammer, who is one of the Insplorion's founders and is now current CTO at the company, is joined by 4 other PhDs. Insplorion's CEO of 7 years now, Patrick Dahlqvist, has vast entrepreneurial and project management experience, having himself founded several successful companies. Nanoplasmonic sensing, or NPS, as well we'll call it for the rest of the presentation, detects changes in the optical properties of the materials, namely the refractive index. Because we track the refractive index, then the NPS is an optical technology. It's real-time, and most importantly, it's label-free and extremely surface sensitive. The sensors that we use usually are composed of noble metal nanoparticles on the surface of a glass support that contain also a dielectric coating. You can see a picture here of the sensors on the bottom left with an SEM image showing, in this case, gold nanodiscs and an actual macro-sized picture of the sensor. Our sensor signal tracks changes in anything that actually effectively changes the refractive index on the surface. It can be its sword mass density or film thickness or a conformational change on the surface.
The physical phenomena behind NPS is the localized surface plasmon resonance, or LSPR. LSPR is a collective oscillation of the electrons on the surface of the nanoparticle. When the light with the right frequency is irradiated upon the nanoparticle, these electrons will oscillate together, creating an electrical field on the surface. The electrical field on the surface will have 2 consequences. The first one is that it will amplify all the modifications that are done on the surface. The second one is that it will interact with the light, which means we can optically track what's going on the surface. This means the noble metal nanoparticles are going to act as optical antennae that can detect changes in the close vicinity of the surface. Now, adding material to the surface of the sensor is going to interfere with the electron cloud and therefore create a shift in the way that the nanoparticles interact with the light. This means we can track things, and that's exactly what Insplorion does. If you had a material like a protein or a film that you deposit on the surface of the sensors, the light is going to interact differently with the nanoparticle, which means we can track it by following something that looks very much like an absorption peak. In this case, is an extinction of the light by the nanoparticle. So by adding a protein or a thin film to the surface of the sensor, the peak is going to shift according to the variation of the refractive index, which means we can track effectively what's happening on the surface. And you can see the peak position shifts as we add material to the surface. By using our software, you can track the position of the peak very precisely. So as you add material to the surface, the peak will change, and the algorithm of the software is going to track the position of this peak, which if you follow in real time, which you can do with our software, then you can see a perfectly good-looking absorption curve. Now, I've been focusing on the peak position because that's the most important parameter for most of the users. However, if you're working with nanoplasmonic interference or with nanoparticle studying, then other features might be important, like the possibility to track the height of the position, so the extinction, or the full width at half maximum, which usually indicates a change in the way the plasmonic behaves as well. Our software allows you to track the peak position with a time resolution of 1 Hertz, and our spectral resolution is in the order of the 10 picometers on the wavelength. This means that NPS data is in real time going to allow you to acquire in situ measurements that will reflect either mass, density, thickness, conformational changes. There are 2 very important features. The first one I've already mentioned, which is that the technology is very surface sensitive, so it will measure within the first 30-40 nanometers above the surface, but not more than that, so it's very bulk insensitive. But another feature that I haven't mentioned is that NPS actually detects dry mass, what this means is that the technology is insensitive to coupled solvent to the molecules that are being added to the surface. And the reason for this is that the measurement is based on the refractive index, and the coupled solvent is usually the same molecules that contain the buffer, so they have the same refractive index. So adding material that is dissolved in a buffer is not going to give you information about the water content by itself, but exclusively the dry mass of the material that is being absorbed on the surface.
So now I will talk a little bit about our instruments. So this is the whole portfolio of Insplorion, including the 3 instruments that we'll briefly go through and the sensors needed to run them. The first instrument developed by Insplorion was the Insplorion X1. It allows studying solid-state reactions at nanomaterials interfaces. So the instrument was designed to study gas-solid interactions. It's very good for catalytical applications, for example, or applications that require to go to a very high temperature, as the instrument can reach 600 degrees without any major problem. And the system is also compatible with mass flow controllers, so you can use different gas mixtures to study how they interact with your material. It will provide you real-time analysis, and it's very easy and robust. That allows in situ measurements, and on request we can also make it so that it can hold 2 samples at the same time. Then there is the Insplorion X Nano, which allows you to monitor dynamic processes in nanoscopic materials and interfaces. The X Nano was designed for applications that require liquid handling. It can also be used for air applications up to 80 degrees, so it has a control temperature up to 80 degrees, and it allowed true nanoscale measurements that is suitable for any sample material, since you can work with gas, with liquids, with solids. And like all the other instruments, allows real-time analysis. It is a complete system and is quite user-friendly. The X Nano will allow you to work with very small volumes with a minimum sample of 20 microliters. It will also allow you to control the temperature from room temperature to 80 degrees without sacrifice of quality of signal, as our sensors are built to sustain such temperatures. And the system will allow you to acquire data at the frequency of 10 Hertz, which means you can get a sample point every 0.1 seconds. The system is built to withstand different materials. It's very resistant materials we used, so you can work with whatever solvents you might require, and our typical allowed flow rate is between 20 and 200 microliters per minute. The Insplorion X Nano's configuration is in transmission mode, which means there will be light going in and going out on the other side, which means the sample will have to be transparent enough for the light to go in, and the liquid flows perpendicularly to the light beam. The third product is the Insplorion Acolyte, which is a little bit special since it is a combination instrument for a QCMD instrument. So the Insplorion Acolyte will allow you to have an acoustic and an optical measurement at the same time. The optical measurement coming from the NPS data and the acoustic from the QCM data. So the instrument then is designed to work with QSense's window module. and it will work very well with either the Explorer or Analyzer, also known as the E1 and the E4 modules. And when plugged together, the system looks like this, and you can see it fits very neatly. The main advantage of doing this is that you can have both acoustic and optical measurements at the exact same surface, so without the need for extrapolations, as you know you're measuring at the same time in real time both techniques. So the optical configuration, due to the way the QCM crystals need to work, has been changed to reflected light instead of transmission. So now we're working in reflection mode. So the Acculite sensors are quartz crystals that will give you an acoustic signal with Insplorion sensor structures on top. NPS complements the QCMD data very well. So other than the advantage of getting the simultaneous real-time measurements with both techniques, you get complementary information from both techniques as well. On one hand, you'll be gathering data from the Insplorion system, which will provide you information about the dry mass and what's going on at the very surface of the sensor. On the other hand, you will have the data coming from the QCMD with information about the wet mass and the viscoelasticity of the material you're studying, and a sensing depth that is much higher. So in combination, you will get both the dry mass and the wet mass, as well as the viscoelastic properties of the material you're studying. But also you will enable depth profiling that comes from having two techniques with different sensing depths. So a combination of the LSPR phenomena, the sensor architecture, and the instrumentation and software that we bring to the table enables unique scientific opportunities where you can combine real-time measurements, triggers of change like temperature, or liquid or gas flows where you can change chemistry, concentrations, pH, as well as light triggers through optical fibers, together with a short probe depth that will allow you extremely sensitive measurements and control over what's happening over the surface. So now I'll go over some of the applications that are possible by using our technology that exemplify perfectly how versatile our technology can be. So I will start with biomolecular adsorption. I've been talking about adsorption of molecules on the surface of the sensors. In this case, we're talking about lipids. So NPS allows you to work in real time and measure mass and conformational changes of lipid structures or vesicle adsorption or deformation and lipid bilayer formation. This is experimental data from Professor Nam Yoon's group, in which he demonstrates that vesicle adsorption from a solution occurs until there's a critical coverage of the surface of roughly 30%, at which the lipids rupture and form a perfect bilayer. This is a characteristic curve that you will observe when studying lipids on surfaces. If you see this, you will know you will have a lipid bilayer that has been formed. Professor Nam Yoon's group has brought the research further and has studied how different surfaces that are available at Insplorion interact with the same vesicles. And you can see in the silica coated sensors, there's a lipid bilayer formation with a standard curve that we were just looking at. But for example, in titania coated sensors, there is only adsorption and not the formation of the lipid bilayer. So the surface influences how the vesicles will distribute or not. In this study, uncoated sensors were also tested, and these uncoated sensors have silica in between the gold structures. And what was observed was this hybrid between lipid bilayer formation, which occurred on the silica part of the sensor, and vesicle adsorption, which occurred on the gold structures. Once the lipid bilayer is formed, you can go a step further and you see how molecules interact with that bilayer. In this case, the protein interaction with the lipid membranes, it was cytochrome C was used to see how it would incorporate into the membrane. In this case, the Acculite instrument was used. So other than the NPS data, which is shown in orange, you can also see the QCM data, which is shown in blue. And you can see there is a big difference between the mass calculated with NPS and with QCM. So this is a good example to see how much water is attached to the cytochrome C that is going into the membrane. I would like to remind you that the NPS only provides dry mass, while the QCM only provides wet mass. So in this case, the difference should be related with the content of water present with the cytochrome C. Another application that now can be extremely important, given the fact that we are always tracking nucleic acid material from viral particles. NPS can also be used to detect DNA. In this case, one would argue that it would work for other nucleic acid types as well. And in this case, the sensitivity is so high that it allowed kinetic discrimination of single base mutations on large pieces of DNA. In this case, what was tested were two different sequences, one that was perfectly matching and one that had a single mismatch. And then the binding kinetics of these molecules to a probe that was immobilized on the sensor surface was measured. One would expect that the matching sequence would perfectly bind to the probe, as you can see with the pink triangles that correspond to 100% of a perfectly matching sequence. And the mismatching sequence, of course, would not be as efficient in binding as a perfectly matching sequence for the same stringent conditions. As one expects, the matching sequence provides a perfectly good-looking binding curve, as you can see with the pink triangles corresponding to 100%, so exclusively matching sequence. But in this case, the mismatched sequence gave such much poorer binding kinetics, so the researchers took it a step further and started measuring mixtures of mismatched and perfectly matched sequence. I'd like to remind you there is only a single base difference between the matching and mismatching sequence, and the difference in the binding kinetics is so high that the researchers took it a step further and were able to distinguish between different ratios of matching and mismatching sequences. So the system is so sensitive that allows for detecting even different mixtures, which can have high impact when studying human genetic diseases, for example. Another application that can have impact on health and drug delivery is the study of hydrogels and their characterizations. Our system will allow you real-time measurement of loading or unloading of a payload into or out of a hydrogel. So you can use the system, for example, to monitor how proteins, in this case it's BSA, interact with your hydrogel or material, be it by adsorbing on the surface or actually going through and into the gel. Or you can use the system to test how modifications to your material would change the interaction with their payload. In this case, a polyarginine peptide was loaded into 3 different gels that have the same gelatin base, and then 2 of them have been modified either with hyaluronic acid or with aldehyde hyaluronic acid that change completely the properties. And you can see the difference of the gels, and the gel that contained the aldehyde hyaluronic acid combo shows much higher capability to interact with the polyarginine peptides. So you can easily test how modifications of your material can allow you to increase the payload, speed up the payload, or load more material, unload faster, unload slower. There's a huge amount of possibilities here. Similarly, one can study the diffusion of material through films, or it could be done the same with hydrogels actually. In this case, it was a little bit more physical application. They were studying organic dye molecules and how they diffuse through mesoporous titania, with the idea of studying them for dye-sensitized solar cells. And the study consisted of comparing how this organic dye was diffusing through different thickness materials. And you can see in blue here how the signal would look like when flowing this organic dye through a 12 nanometer compact titanium surface, as is exemplified on the left. So of course there isn't much space for diffusion. The film is compact, so there will just be adsorption at the surface. And of course, given the time for the organic dye to reach the surface and absorb, you have an almost immediate absorption curve. In comparison, you have the red graph, which corresponds to 10 micrometer mesoporous titanium film. And I'd like to remind you that we're only sensitive on the first 30 nanometers closer to the surface. So of course, if you have a much thicker film, even if it's porous, it will take a longer time for the dye to reach the surface. So of course, the response time is closer to the hour mark rather than just a few seconds. So this is a great example to show how you can use the fact that we're very surface sensitive to your advantage. In this case, because we were using an organic dye that has its own absorption peak that doesn't overlap with the plasmonic peak, because the dye has this color and we're tracking 525 nanometers, which is the peak of absorbance for this dye, we know that after the 8th minute, the dye reached the measurement chamber of the system. And you can see this by the increase in the green graph that is very quick and very steep. But you can see that it keeps increasing over time, which gives you a hint that it might be penetrating through the film until it reaches almost the saturation. And curiously, once the dye concentration starts to stabilize is when you start having the plasmonic peak change, is when the NPS signal starts to kick in. And this makes sense because once the solvent starts to stabilize, it means the film is becoming saturated, and of course that's the time when it's reaching the bottom of the film, so exactly when it's reaching the surface of the sensor. So of course that's when NPS is more sensitive, that's when we start getting the signal, and of course the signal stabilizes fairly quickly, and once both NPS and the dye cannot go any further, they're both stable, the dye is removed, and of course whatever dye was in the chamber that was not incorporated into the film gets washed away, and you can see that looking at the green curve again at roughly 35 minutes, but then also the NPS signal starts to go down, which means without the presence of the dye in the solution, the dye starts diffusing away over time, which is something you can also measure with NPS. Of course, the researchers kept going and studied a bit further, so they varied film thickness and they varied dye concentration, and with this information they were able to get quantitative kinetic data that allowed them to calculate the diffusion coefficient and the concentration that was being retained on the films during the experiments. So this is a great example on how to quantitatively use the NPS data, but also on how to explore the information that is available. On a completely different note, but I thought it would fit the topic of health post-COVID because it relates to pollution, and of course pollution is one of the major health factors. Insplorion technology also allows gas studies, and in this case the study of gas adsorption in microporous films and determining heat of adsorption. In this case specifically, CO2 was used with a specific polymer using the X1 instrument. So once again monitoring the peak position, but also the absorption band of the polymer that was spin coated on the surface. The researchers used time-resolved NPS and its response to calculate adsorption isotherms, and they did that also while changing the pressure of CO2, the partial pressure of CO2, but also the temperatures. And a combination of all these allowed them to calculate the isosteric heat of adsorption, which is a very good indicator of how a gas is being adsorbed to a material. In this case, the result was in excellent agreement with the literature and shows that the material was good for capturing CO2. So this is another way you can use this fantastic tool that is NPS. The range of applications that NPS enables is actually much broader than the one I've just told you about. If you want more information, or if you want to have a look at more options, please come to our website and check our application areas page, where you'll find this interactive tool where you can choose what kind of materials you're looking at, what kind of processes you're looking at, and once you hit the combination, you will find publications, application notes, reference customers that have been doing work in that area. If you find a combination that doesn't have any information on it, it's because no one has dared to do that. So please talk to us, discuss with us whether it will be feasible to work on your idea or not. We are here to help, of course. And on that note, I'd like to thank you for your time and say that if you have any need for more information or if you have any questions, please send me an email. I'm always available, or someone else from the Insplorion team will be. You can always visit our website at www.insplorion.com, and we'll be glad to receive a contact from you or a visit. Thank you.